Dear learners, now in this section we shall study anti-inflammatory role of spices and we shall also study physiological effects of spices and bioactive compounds. Now, chronic diseases also called as non-communicable diseases that include Alzheimer's disease, arthritis, cancer, cardiovascular diseases, diabetes and Parkinson's disease remain the primary root cause of death and disability worldwide. The major risk factors associated with these diseases are unhealthy lifestyle including lack of physical activity, poor diet, stress, excessive tobacco and alcohol consumption, exposure to radiation and infection with pathogenic microorganisms. It is now well established that these agents include inflammation and dysregulate inflammatory pathways which lead to the development of chronic diseases. Inflammation, which means to set on fire, is a body's natural response against harmful pathogen and stimuli that occurs in two stages, namely acute and chronic inflammation. Acute inflammation is a part of innate immunity initiated by the immune cells that persists only for a short time. However, if the inflammation continues, the second stage of inflammation called chronic inflammation commences, which instigates various kinds of chronic diseases including arthritis, cancer, cardiovascular diseases, diabetes and neurological diseases via dysregulation of various signaling pathways. Eventually, it was discovered that transcription factors such as nuclear factor kappa B, signal transducer and activator of transcription 3, that is STAT3, inflammatory enzymes such as cyclooxygenase 2, matrix metalloproteinase 9 MMP9 and inflammatory cytokines such as tumor necrosis factor alpha TNF alpha, interleukins that is IL such as IL1, IL6, IL8 and chemokines are the main molecular mediators. Numerous results from preclinical and clinical studies over the last several decades have ascertained the efficacious role of spices and their active components in preventing and combating various diseases including arthritis, asthma, cancer, cardiovascular diseases, diabetes and neurodegenerative diseases. The most commonly used spices for culinary purpose that shows biological activities are black pepper, cardamom, cinnamon, clove, cumin, fenugreek, fennel, garlic, ginger, onion, rosemary, turmeric, etc. Now, let us study the physiological effects of active components of spices. Increasing lines of evidence have established the efficacy of principal components of spices in preventing as well as alleviating different types of chronic diseases. The main components of spices and their curative potentials are now discussed. Now, let us study the potential mechanism and physiological effects of bioactive compounds one by one. First is 1,8-cineol. It is a monoterpene oxide found in variety of spices such as basil, cardamom and sage. Cineol has been used to treat multiple inflammatory disorders such as bronchitis, sinusitis, chronic rhinitis and asthma. In addition, numerous studies also showed its potential in preventing different chronic diseases such as asthma, colitis, chronic obstructive pulmonary disease, pancreatitis, etc. by modulation of inflammatory pathways. 6-gingerol, the main active component of ginger, is shown to possess different biological activities such as antioxidative, anti-inflammatory and anti-proliferative properties. Its therapeutic effect was observed against various chronic diseases such as Alzheimer's disease, colorectal cancer and diabetes. Studies have shown that this nutraceutical is a potential candidate for the treatment of diabetes. Moreover, 6-gingerol possesses anti-tumorigenic and pro-apoptotic properties and alleviate symptoms of allergic rhinitis. Alpha pinene is a monoterpene found mainly in eucalyptus oils and oils of aromatic plants such as rosemary. It is known to possess antimicrobial, apoptotic, antimetastatic and antibiotic properties. Alpha pinene is one promising agent for the treatment of various inflammatory diseases. The inflammation associated with acute pancreatitis is considerably reduced by treatment with alpha pinene in vivo and is also anti-allergic agent against AR. Now, next is garlic. Diallyl sulfide, that is DAS, or allicin. It is the major organosulfur compound of garlic. It is a potential agent for treatment of airway inflammation such as asthma. Diallyl sulfide has also been demonstrated to have anti-cancer properties against different cancers such as colon cancers, prostate cancer, skin cancer, etc. via modulation of inflammatory pathways. Curcumin, an active component of turmeric, is the most widely studied nutraceutical. It is known to possess antioxidant, antibacterial, anti-cancer, antifungal, anti-inflammatory and antiviral activities. Thus, it is a potential agent against various chronic illnesses. Furthermore, curcumin was shown to ameliorate the insulin signaling in the brain of Alzheimer's disease in vivo, thus showing its feasibility for treatment of Alzheimer's disease. This compound is known to inhibit cancer cell proliferation, survival, invasion, angiogenesis, metastasis, chemoresistance and radiation resistance in different types of cancers via modulation of different signaling pathways including nuclear factor kappa B. Curcumin has been shown to inhibit inflammatory bowel diseases, colitis, rhinitis, oral lichen planus, psoriasis and prostatitis in various clinical trials. It has also been shown to inhibit cancer alone or in combination with standard chemotherapeutic agents in many clinical trials. So far, curcumin is the most extensively studied spice derived component for the treatment of different chronic diseases in both preclinical and clinical settings. Diosgenin is a bioactive compound obtained from the spice Trigonella phonium graecum or fenugreek. Over the year, this spice has been known for its anti-carcinogenic, anti-diabetic, antioxidant, hypocholesterolemic and immunological properties. Because of its anti-inflammatory activities, diosgenin is a potential agent for various chronic diseases including Alzheimer's disease, breast cancer, chronic myeloid leukemia and osteoarthritis. Additionally, diosgenin was found to exhibit antiviral activity against hepatitis C in vitro, induce apoptosis in hepatocellular carcinoma and prostate cancer and inhibit migration of human breast cancer in vitro. Now, next is capsaicin. Capsaicin is a principal component of the spice red pepper or capsicum. It is highly efficacious in ameliorating several chronic diseases such as asthma, diabetes, cancer of breast, cervical, stomach, etc. Additionally, capsaicin also exhibits anti-cancer activity against cancer of colon, lung, prostate, skin and tongue. In vitro and in vivo studies also revealed that capsaicin ameliorated chronic diseases such as Alzheimer's disease, skin inflammation, small cell lung cancer, etc. Now, the next is Eugenol. Eugenol is the active principle from clove extract, well known for its anti-inflammatory properties. In addition, it has been shown to inhibit various chronic diseases in preclinical studies. For instance, Eugenol was shown to restrict the progression of asthma in vivo by inhibition of nuclear factor kappa B NFKB pathway. This compound also inhibited cell proliferation in gastric cancer in vivo by suppressing nuclear factor kappa B pathway. Eugenol was found to enhance the efficacy of anti-cancer drug gemcitabine and exert anti-inflammatory activity in human cervical cancer cells. In addition, Eugenol was shown to inhibit skin cancer. Numerous investigations further revealed that Eugenol exhibits antidepressant as well as anti-diabetic activities. The next is cinnamaldehyde. Cinnamaldehyde is the active component of spice cinnamon. This component is widely known for its antioxidant, anti-tumor, cholesterol lowering and immunomodulatory properties. Cinnamaldehyde exerted its anti-inflammatory effect in gastric inflammation by inhibiting nuclear factor kappa B activation. Cinnamon can also reduce allergic encephalomyelitis in vivo via regulatory T cells. Cinnamon bark has a prominent action in reducing inflammation in arthritis model in vivo, hence may be regarded as a potent anti-rheumatic agent. Moreover, cinnamon is also effective for the treatment of neurodegenerative diseases such as Alzheimer's disease. Now, the next is Carcinol. Carcinol is a polyisoprenylated benzophenone isolated from the plant Garnecia indica, that is kokum. A functional investigation has revealed the anti-carcinogenic, anti-inflammatory and antioxidative properties of Carcinol. Studies showed that Carcinol inhibited the proliferation of breast cancer cells in vitro. This active component also inhibited inflammation associated colon carcinogenesis in vivo. Additionally, it also sensitized breast cancer cells to chemotherapeutic agents. Now, the next is Thymoquinone. It is isolated from the black cumin or known as Nigella sativa. It also prevented inflammation, neoangiogenesis and vascular remodeling in asthma in vivo. Thymoquinone also inhibited TNF alpha induced inflammation and cell adhesion in rheumatoid arthritis, thus making it promising anti-inflammatory agent. Studies have reported the ameliorative activity of Thymoquinone against oval albumin induced allergic conjunctivitis in vivo. Additionally, it was also found to be effective against cancer of bladder, lung, ovarian, gastric, etc. Now, the next is quercetin. Quercetin is a dietary flavonoid obtained from onions. The anti-cancer, anti-inflammatory and antioxidant properties of this phytochemical are demonstrated by numerous studies. Quercetin is effective against various chronic diseases including arthritis, breast cancer, dermatitis, diabetes, inflammatory bowel disease, hepatitis, psoriasis, etc. due to its ability to inhibit the dysregulated inflammatory pathways involved in these chronic diseases. Pepperine. Pepperine is the principal plant alkaloid isolated from black pepper and long pepper. Pepperine has several biological properties including analgesic, anticonvulsant, anti-tumor and anti-inflammatory activities. Several studies have shown that pepperine could attenuate the inflammatory response associated with chronic diseases such as Alzheimer's disease, asthma, arthritis, chronic gastritis, endometritis, Parkinson's disease, etc. Now, sulforaphane is an isothiocyanate, that is sulfur containing compounds distributed amongst cruciferous vegetables including mustard. Studies have shown that sulforaphane possesses anti-cancer and cardioprotective activities. It elicits protection against cardiovascular diseases via activation of NRF2. Studies also reported that sulforaphane represents a promising agent for the treatment of chronic diseases such as Alzheimer's disease, bladder cancer, colorectal cancer, diabetes and lung cancer. Allicin in garlic, anethole in fennel, carnosol in rosemary, linalool in coriander, crocin in saffron, sesame seeds, ursolic acid in basil, carvone in mint and myristicin in nutmeg found to aid in preventing and alleviating various chronic diseases mostly by down regulating signaling pathways. So, with this we have finished the physiological effects of bioactive compounds in spices. Thank you.